All right. Okay. Good evening, everyone, and welcome to Genomics Lite. This is our first session for this academic year. My name's Em, and I'm sort of hosting the session, keeping it running, and doing all the magic Zoom stuff on this end. And we've got a guest speaker along this evening, but I'll pass over in a little bit, and I'll let you introduce yourself in a second. Fabulous. We've got quite a mixed audiences tonight, which is always nice to see. So, I think, having said that, I will stop sharing. And I will hand over.
Hi everyone, I'm Sherry. I'm an advanced research assistant at the Wellcome Sanger Institute, and I'm going to be talking to you about sequencing, more specifically long read and short read sequencing, and we'll have a nice little discussion about that. So just kind of like beginning, like who am I? So as I said, I'm an advanced research assistant in the COVID-19 genomics surveillance. So what we do is basically monitoring the strains of COVID and reporting that back to the government. I also do data analyzing, not very fun, but it's also the official title is called psychometrician. So I'd be working mainly with kind of like databases and data points, something like that. So I have a bachelor's in medical genetics from Swansea University, and I'm currently doing a master's in genomic medicine at King's College and St George's University. And if the interviews go well, hopefully medicine next year as well. So we'll see about that. And I'm also a CoSTEM ambassador. Now just kind of like a starting point. In most things like maths, if we have a lot on the x-axis like time and then the y-axis like not difficulty, in an ideal world, we want the difficulty to, increase steadily in a linear relationship. But in genetics, this isn't the case. Actually, what you get is something like this, which is an exponential curve, where as you go on, it gets quite easy, say DNA bases, alleles, blah, blah, blah. And then all of a sudden, it gets really, really confusing. So hopefully my talk isn't confusing. But if it is confusing, make sure you ask lots of questions, because that's why I'm here. To answer all those questions. So don't worry, I will let you know which bits are a bit, kind of ahead of kind of A levels and like what sort of area or what sort of level this is. So don't worry too much as well. So today we're going to kind of try and be able to understand the terms gene expression and mutations. We're going to learn briefly about sequencing methods, so mainly Illumina sequencing and also the long-read sequencing of PacBio, Oxygenable. Look at the uses, the advantages and disadvantages, and then kind of introduce some terms to you, such as sequencing by synthesis and reference genomes. So hopefully you're not going to be experts by the end of this, but hopefully you'll be able to see these words and terms, and when you do see them, you'll be a bit more familiar to them. So this is actually a slide that I give to year sixes. What is DNA? And I always use this slide because I think it's very, very applicable right now. DNA is this structure, and it's the book of information. It tells us everything about us in a weird, funny language. Now, if we were to go in a bit more in depth with that, this is kind of the more A-level, sort of level. Is DNA is actually a lot more than just a book of information. DNA stands for deoxyribonucleic acid, and its kind of building blocks. So what it's made of is called nucleotides, and they have like 3 components to it. So that's the nitrogenous base, which is just, your A's, your T's, your C's, your G's, and you have your phosphate group, which is, if you can see my mouse, but these kind of P's here, and you might have heard the term, you might have heard teachers say the sugar phosphate backbone, and that's made up of the sugar, which is in the DNA's case deoxyribose in your phosphate group, and then this red square kind of indicates a nucleotide. So that's when anyone ever says nucleotide, that's kind of what it means. So going over the basics here, we just want to understand kind of these basics. So when we do get to sequencing, we're really understanding those types of points. So just as a quick quiz, can anyone tell me how many base pairs there are in the human genome? Just by my guess.
So there should be a poll on your screen right now. If for whatever reason it's not showing, you can pop an answer in the chat box as well. Awesome. Quite a mix, but we'll give another minute for everyone to have a vote. Okay, quite a split of answers, and actually quite a lot of people. Actually, there's sort of votes for all of them, but the top 3 votes have gone to 3 billion, 20 billion, and 6 billion. So the smallest and biggest numbers were not voted on. So somewhere in the billions is the consensus.
That's a very good guess, and so actually the answer is. I think it is 3 billion base pairs. So that's 6 billion bases. So it's a bit of a trick question because you can remember like 6 and then actually it's 3 billion base pairs. So a lot of the time we do that a lot in genetics. But yeah, it's 3 billion. It's a hell of a lot of stuff. And in some ways, this kind of introduction bit might be a little bit basic for everyone, but it's just to understand kind of how is DNA packaged and all that stuff. Because once you understand that, you can really understand how we can read them, how we can sequence them. Just a little bit more about DNA structure. So as we know, there's DNA and also something called RNA, which is prevalent within viruses mainly. So just a quick reminder on the right side, our DNA bases, so those nitrogenous bases I was talking about, it's 4 of them. So we have our adenines, so A's, thymines, T's, cytosines are C's, and guanines are G's. Now, A's and T's go together, and C's and G's go together. Something that is important in the terms of sequencing is A's and T's have 2 hydrogen bonds, and C's and G's have 3 hydrogen bonds. And the reason that is because hydrogen bonds, they cost energy to break them. So if you have more hydrogen bonds, it's going to cost more energy to break them. So that's why you have to take that into account sequencing. The one big difference between RNA and DNA is obviously the fact that it's single-stranded and not this beautiful double helix shape. And also one of the bases is changed. So instead of thymine, you have a uracil, which is a very small structural change, but otherwise it's the same. I think it's just missing CH3 group. Yeah, that's DNA structure. But like we said before, there's 3 billion bases, and that's within 3 billion base pairs, within every cell of our body, apart from your sperms and egg cells. But so how is this all packaged? We need to know how that's stored, and that's how we're looking now. Is the DNA is wrapped around these very tiny proteins, and they're called histone proteins. These proteins are the smallest we have in the body, so they're so small, in fact, I don't even think we can really see them unless, I don't even know how you can see them anymore because you can't even see them on a microscope. So I'm not even sure how scientists found that out, but they're incredibly tiny nonetheless. And they kind of come into this, like what we call optimer structure. So there's 8 of them that kind of bunch together, and that's how the DNA will wrap around it. And then it will wrap even more to create a supercoil. And then all of that kind of packaged material is what we call a chromosome. So when people say chromosomes, it's just very tightly wound DNA around proteins, really. And that's what we got DNA, chromosomes. So just another quiz, just so we can see kind of the baseline level is, how many chromosomes do humans have?
Awesome. So again, we've got a poll on the screen. I'll give you a few minutes to make a vote. All right, I've got an almost unanimous vote this time. So a couple of other answers, but I think it's like 80% of people are gone for answer A.
Yeah, they would all be very much correct. It's the word. Yeah, it's 46 chromosomes to 23 pairs. So when I mean gamete cells, I'm talking about your sperm and your egg cells. They're what we call haploid. It's a term. It just means they have half, because that's why you get one from your mum, one from your dad, and you get 46. But the rest of the somatic cells, so other cells that will have DNA in them. So your brain cells, nerve cells, they'll all have 46 chromosomes. But yeah, that's brilliant, fantastic. We know that. So I guess another point is like, why is DNA important, and why are we even bothering all this? It really is just a bunch of letters, and a lot of the time, scientists don't even understand 70% of them. So why are we even bothering? And the big reason is this idea of what we call gene expression. And that was one of the terms we wanted to kind of understand by the end of this talk. And gene expression, in simple terms, is using these letters to make protein, and then these proteins can help the body in some way. So some genes will be encoded. So genes are, in some way, just a bunch of letters, and it can be from 7 to 1,000,000. And when we take those letters together, they can make a protein, and that protein can help. So that could be like an enzyme, or it could be something else like that. So the basis of that is every 3 letters makes what we call an amino acid. Now I'm more than certain everyone knows what amino acids, but I'll just go over it one more time just so we're all on the same level. So an amino acid is the simplest form of a protein. So protein is built from a chain of a polypeptide of amino acids. And to even go beyond the amino acid, you need 3 bases, and these 3 bases are also called triplets. Now you can have many different variations of letters to get the same amino acid, but yeah, we only have 20 amino acids, so there's not many variations of that, and that's what we call gene expression, is taking those letters and making amino acid to then make a protein. So actually, genes, I probably didn't do that good of a example of explaining what genes were, but the National Institute of General Medical Sciences does a much better definition, which is a gene is a segment of DNA that contains instructions for building one or more molecules that help the body work. So kind of coming back to the enzyme example, you could have a gene that could be this long, and when I read it, it will say make this enzyme, and the enzyme could be lactose, and that lactose is in dairy, so that's how we break down dairy. But some people that gene might not work, or that there might be some of it missing. And then when it tries to read it, it says, oh, I can't make lactose. So that's why some people are lactose intolerant, is a faulty gene because it can't produce protein that it wants to. So hopefully that kind of makes sense. And if it doesn't, I'm sure I can come up with another analogy. Scientists originally believed we had 3 million genes or something like that, because, we must be, because we're so smart. And, I think a strawberry has 50,000 genes or something stupid like that. And so they thought, oh, humans must have more. But actually, in reality, we only have around about 20,000 genes. Men maybe have slightly less. Does anyone know why men would have slightly less genes than women? Just like if you think you know the answer, just type it in the chat box.
We've got an immediate answer, very succinctly put, because the Y chromosome is slightly smaller than the X chromosome.
Yeah, that's literally, yeah,
perfectly put.
So actually the X chromosome is actually much bigger than the Y chromosome. There's not a slight difference. It is much bigger, just as a slight correction. But yeah, that's the answer. Is the X chromosome is much larger, and it has holds a lot of lots of genes on there. Not to say that that the woman has way more genes because it's got 2. Actually, the concept in genetics is called an inactivated X chromosome. So actually, what you get is when you have a female, so a woman, you'll have one that kind of works, and then the other one will mainly turn off, but some of them will be on. So that's kind of how the genes balance out. So it's not a massive difference between genes between men and women, but it is a slight difference. So actually, in our DNA bases, only about 25 to 30% of the human genome have genes in them. So that's not a lot. That's only I can't even. I'm not very good at math, so I'd have to get a calculator out. But that's whatever 3 billion base pairs, times 30%, really. So that we only know that many, really, how much is. We're not really sure what the rest do. We have a vague idea, might to do with rRNA or transposons or things like that. Those are lots of terms that you may have to Google if you're not quite sure about them. But yeah, scientists aren't really too sure about what's going on, and that's kind of why we think sequencing could be that way of being able to find out what those other stuff does, because it definitely does something, not just nothing, but it's not genes, and we know that for sure, certain. So, so another thing that I wanted to, out of interest, does anyone know what percentage of the genes, so out of those 20,000 genes, are actually protein encoding genes? So just to clarify that question, so some genes might be for something else, like maybe helping the brain move or something, whereas another thing will be to make specifically that gene makes a protein.
Got some very quick voting going on here, and there's a pretty good consensus for answer D of 1 to 2%. I've got about 50% of people going for that, a smattering for the others. Nobody's gone for 0, which is good, because logically we'd end up with no proteins if we had 0%, but most people are going for 1 to 2%.
Yep, that's perfect. It's only 1 to 2%, which I found insane when I first thought. I was like, that's what the hell. But you think, what does the rest do then? But yeah, it's only 1 to 2% of human genes actually encode for proteins, which is somewhat helpful, because it means that if there is a faulty protein, we actually know we have a much lower scope of finding out where the problem is, because we know there's less genes to look at. But it's still insane. It just shows that scientists don't really know much about the human genome. And this kind of brings over to this picture here. The year 6 slide that I showed at the start with, DNA is a book of information. It's a valid slide. It still makes sense, but it's far too complex, far too simplistic. Actually, in reality, the human genome is much more like this picture, like this library, where a book is rather a gene or a part of the genome, and this library is the human genome, and it's so complex. And if you were trying to find a book in this library, it'd take you a long time, unless you had someone to show you. And it's the same with the human genome, which takes a very long time to look through all this stuff. And a lot of the time, we don't really know what we're looking for or where it is. So this is kind of a much more accurate kind of depiction of what the human genome really is. So kind of going back to this term gene expression. So what is gene expression? So I'm not really going to go through transcription and translation, because this is very much A-level kind of territory. So I'm not going to go through that, because I know AQA, OCR, EdXL, they have their own steps that they want you to say, and you get marks for them. So we're not going to go through that. We're just going to go through a very kind of basic concept. Now, like I did for my A-levels, is if anyone watches Khan Academy, they're going to go through this concept called the central dogma. And it's a very important concept, and it's essentially how you go from a DNA to a protein. And to do that, we need 2 processes that let us go from that, and it's transcription and translation. Now, the way I like to think of transcription and translation is just using an analogy. So transcription is essentially, so if you think about it logically, you've got to go from the nucleus where the DNA is to the cytoplasm, where you've got to make your protein, and then that protein needs to move around the cytoplasm. So it's got to be transported and then changed into something else. So transcription is the step where we're transcribing our DNA into RNA. So in other words, imagine if I had a piece of paper that's very fragile, and if I need to run to another building, and it's absolutely chucking it down. If I'm going to run to that other building, it's going to get very, very wet, and it's going to be hardly readable. So if I transcribe that onto, let's say, a laminated piece of paper that's waterproof, I can then jog to the next building, and my thing will be fine. I can just wipe off the water, and I can read exactly what I've written. Whereas translation is much different. We're taking our RNA, so that laminated waterproof paper, and we're turning it into something else, so into protein. So in that analogy, I'd say, you imagine you have someone French and they only speak French, and they want to read what you've written. You're going to have to translate all of that information from that into French. Now, I wouldn't be able to do that, because I'm terrible at French, but the concept's still there. You're transcribing and translating, and that is the really what gene expression is. You're going from DNA to protein from the gene that they've read. So hopefully that makes sense to everyone. So this is kind of where sequencing and clinical geneticists have their fun as well, because what happens when it gets wrong? We get something called mutations. Now, when I hear the word mutations, I think, wow, that's really bad. Now, I don't know what you guys think. Do you guys think it's good or bad? I think it's something that whilst I'm going through this slide, just kind of think to yourself, is it good? Is it bad? Is it both? Is it nothing? Just have a think about what you think a mutation really is. Now, a mutation in simple terms is just changes in the DNA bases. So I've got a few examples on the right-hand side where we can have substitution. So a substitution is just swapping one base for another. So for an A for a T, that could do nothing. Like I said before, there's multiple triplets of multiple things that can kind of make up amino acids. So maybe the one I've changed actually makes no difference to my amino acid. Maybe the amino acid does change, but it doesn't affect the protein folding of my protein. So actually there's nothing wrong with it. Or it could affect it. We get into a much more kind of different kind of territory when we talk about deletions and insertions, because that kind of does mess with everything. So they do tend to be something called I can never pronounce the word, but deleterious. Deleterious, whatever. You know what I mean. That's when it kind of goes into that territory. So these are, you kind of would start to think they're bad. But it's kind of rhetorical question: is it good or bad? The answer is, well, it's actually both. It is good and it's bad. So it's good because genetic mutation. So every single person will have the genetic term is single nucleotide polymorphisms, or SNPs, which is the slang. But that just essentially just means that if I was to take, let's say, M's genome and my genome, and we were to compare them, they wouldn't be exactly the same. Not because that we're male and female or difference like that. Even if I was to take someone that was similar to my race and same gender, or even someone from my family, if I had, we would have similar genomes, but not exactly the same. And that's why we get genetic variation. That's why we get people look different, people look a certain way, etc. So yeah, mutations are important because, if not, we'd all just look the same. And also it's important in biological evolution. So I think most of you may know this, but I'll say it anyway. So 3 billion years ago, mitochondria actually we had a single-celled organism, and mitochondria essentially fused with that single-celled organism. And since then, we have lived together, and that's kind of why we have them. So mitochondria actually have their own DNA, called mitochondrial DNA, much smaller than ours, but they still have some. So that's how we kind of know that it was definitely a foreign being, or wasn't always a part of our cells. And actually, we can tell through testing that the genome has actually been selected. So like kind of Darwin's principle, where it's been like selected slowly. So meaning that genes have been cut down or got rid of. So now I think there's only like maybe something like 16,000 base pairs or something like that. Not many base pairs in the whole in mitochondrial genes, and it's something like 32 genes, whereas before there were hundreds, like maybe thousands of years ago. So it's definitely, sorry, not thousands, millions of years ago. So evolution has occurred through kind of mutations and saying that we don't want this one, we want this one. So they are good in helping us in the long run. There's more examples of biological evolution, but don't really have time for that. But the more common times what you hear is the bad ones. And that's where we get things like genetic disorders, so like Down syndrome, so that causes from mutations. You could also have cancer, which is a very, very popular sort of answer to give when you think of mutations. You think, oh yeah, radiation and skin cancer or some stuff like that. So they can be good, but they can also be bad. So it's good to kind of remember both and see that mutations are good and also bad. So why does it happen? I think that's a very important point to remember. It's difficult to say what, but we have external sources as well. So it's not just from from mistakes within, it's also from outside. So like I was alluding to before, with skin cancer, we have UV radiation, also X-rays, which is why I think pregnant women are normally told to make sure that they know that they're pregnant before they get an X-ray. Alcohol, smoking, fatty foods, things like that, we all know. And I think as well, you may have heard, I think it was a GCSE topic, the thalidomide, it caused deformed babies and actually affected future generations because of the way the drug interacted. And for those that don't know, thalidomide was a drug that was meant to help with morning sickness, but actually it was, in some ways, if the drug was meant to be like this, it was like that. So it looked the same, but it was the opposite way around, and it's called a racemic mixture, if you ever want to look that up. And that actually, the opposite version caused a detrimental effect, which caused deformed babies, and I think a lot of them did actually die as well. So so external components do have a very important nature, and they can actually affect your future generations. And a big thing is epigenetics, which is where you may be born with what your parents passed down, but actually, as you've gotten older, your lifestyle can affect what genes are turned on, off, and that can also affect your future generations as well. So external agents have a big thing, and COVID as well, long COVID. So I could go on and on and on about this. I need to stop, but the point is there that external stimulants have a very big play also affecting mutations. Now, internally, we have DNA replication. So even though there are systems to kind of check and make sure that there aren't problems there, there's kind of a lot of kind of problems with that. So we have like translational or transcriptional modifications which don't happen, which I won't go into a bit more because you kind of need to know a little bit more about transcription and translation to understand that. But nonetheless, that is still a point. DNA replication. Ones that you might understand a bit more is in meiosis or mitosis, where we have problems there, and that can cause what we call Down syndrome, which I'm sure everyone's knows, which is called trisomy 21 in genetics terms, which is where you have 3 chromosomes on chromosome 21. And you can get other ones like trisomy 18, which is, I think, T-Turners or something like that. And that all happens from like a mistake within meiosis 1. Or it can also happen in meiosis 2 as well. But it's to do with the non-homologous recombination, the failure in that. So there's a lot of kind of reasons why it could happen, and we're still kind of understanding more things there. So I guess like why is this important is the big thing is, and now this is something that someone put on their slide for my degree, and I didn't understand what they were saying. They say clinical geneticists rank mutations depending on their predictability on being pathogenic. Probably some of you do understand that. If so, you're smarter than me 5 years ago. However, what that means in layman's terms is it just means that if you know how bad a mutation is, you can kind of predict if that mutation will be disease causing. So if I take, that's why all these slides were to show you that actually we need to understand those basics and understand what mutation is and why they're bad, how do they affect us, before we can start even thinking about reading stuff. Because if we read stuff and if I read about physics and I have no idea what I'm reading, there's no point in reading it. So we need to learn that stuff first before we can move on to kind of the reading of things and interpreting. So this is a thing that, this is kind of a pyramid of kind of the hierarchy of different mutations. Now, these words may not mean anything to you, or they may do. Just means that the bottom one means it's also dubbed a silent mutation, which is what I was saying before, where there might be a substitution and it doesn't actually affect the protein or the amino acid, but there is a base change. The second one just means there is a change in the amino acid / protein. And then the third one is kind of the one that you want to look out for. Really, it makes a big kind of difference. It more or less stops the gene itself or gives it a new function, which is not what we want. So we can use this and look at these mutations to try and see what sort of problems some person may have or predict whether they could have something or not to their children. So sequencing essentially is like what I'm saying is taking that library of information, that analogy that I was saying before, and looking for those mutations. So these were the mutations that I was talking about before. So these is a bit more kind of in-depth different mutations. So insertions, deletions, very self-explanatory, deleting a bit or adding a new bit. A repeat expansion. So that is a bit complicated, but if you were to Google Huntington's disease, it is called a CAG repeat disease. So that is essentially where if I have loads and loads and loads and loads of CAGs and you have like, if I have like 30, I'm okay. But if I have like 50, I might get Huntington's disease. So that's essentially what it means. Repeat expansion. There's many more as well. Nonsense is where you have a premature stop codon, so where the gene stops before it should. So if it should have stopped at number 10, it stopped at number 5. That's not necessarily very good. Sometimes it doesn't make a difference, but sometimes it does. And then missense is what I was saying before, where you change the base and it changes the protein. If anyone has any questions, by the way, I want to stick in the chat box. And this is kind of where it's getting a little bit more, like picking up pace a little bit. So, but if you're happy with it, then I'll just carry on and kind of move forward. So short read sequencing is essentially where you take, if you have your gene as the book, you're sequencing page by page, and you take, and that's what we call short read. It's very small fragments or pages or page by page, and then we kind of group it all together to try and find out what the book is saying. That's kind of the way I like to look at it. So hopefully that kind of is useful for you. So the Illumina is something that if you've read it or seen anything about sequencing, is like a massive company. I think they have like a monopoly on everything. Like all the short reads, like all of these pictures here, all the short read sequences, they're all owned by Illumina. So they have a pretty good monopoly on them. But there's a reason for that. They just have a very high accuracy and very good machines. So the ones we have, the ones I use predominantly at Sanger for COVID-19 surveillance is this one. It's called a NovaSeq 6000. Anyone wants to know what that is? You can Google it. But so kind of a good kind of outlook of looking at any kind of sequencing thing is looking at the pipeline of it. So that's just a term what we use by kind of just like what we go from start to finish. So sample prep and the thing is though, is it's very annoying because I can't go into too much detail on all of them. I could probably do a whole talk on each of them separately, but unfortunately I can't. So I'm going to try and skim as much as I can, but just please note that there is a lot more to it and it's not as simple as I'm putting it. Hopefully it makes sense though. So the sample prep is you're just taking your DNA, adding the bits that you want to it, make sure it's identifiable, make sure that I can actually know whose is whose, what's what, make sure it's clean and not actually clean, but make sure there's no other bits that I don't want. I want the bit that I want to check. And once that's all kind of done, which is probably takes about a day or two normally to sample prep stuff, then it's kind of ready for sequencing. It will then be put in the sequencer and we get these really big files. They're like 450 gigs, I think we get from our NovaSeq. It's very big data. It's and they're known in called a FASTQ file format. Just a quick note, by the way, a lot of this is you're not going to need to know. This is all just to this is all just kind of just for your own kind of learning. So if you do want to search in this up or ask any questions at the end, you won't have to, but you're not going to like need this fail level or anything like that. So but this is I would probably say third year bachelor's, maybe even after that. So it's pretty high level. So don't get like too hung up if you don't understand it or it's kind of going over your head a little bit. So once we get those like raw sequencing files, we need to align to a reference sequence. Now, does anyone know if does anyone know why we might need a reference sequence or like what it even is? Anyone's like in the chat box, I'm sure we get some good answers. I can't see anything in the chat, so I'm just going to just answer that question. So really, if, like I said before, we're not really sure what we're looking for, what is what. So if we're sequencing something different that we don't know, or something like that, we need to know that there's no mistakes. We need to be able to compare it to something. So what we're doing is we get, when we're sequencing this new thing, we need to know where's the mutation, where what. So we have something called a reference genome. So a reference genome is not always right, and actually there is a little bit of a debate in genetics of, well, like, which reference do we use? Because I know in humans we have maybe 30. I can't remember. I don't quote me on the number, but it's something like we have multiple. We have several human reference genomes that people use, but unfortunately they're all Caucasian, and as we all know, is different ethnicities and races have different genetics, different sort of kind of reactions to things. So there was a big one with COVID where I think people from BME ethnicities were reacting to the COVID more harshly because of something called a cytokine storm. And that was because of their genetics and the way they do it, and people thinking there's kind of misrepresentation there. That's another topic, but that's something to kind of bear in mind with reference schemas. They're not always perfect, but they do give a very good indication of what we're looking for, and they kind of help us, kind of guide us, like their librarian. They kind of guide us around the library to make sure we know.
We've got 3 ideas coming in the chat box just as you were explain that bit. So someone's put to confirm and compare sequences, which is sort of what you were saying, that it helps us check we are where we thought we were, and you alluded to there, like comparing, like seeing the differences. I like the idea. We've had one about making sure it's the organism that we're interested in, because I guess like in some cases you might have thought you sequenced one thing, but you do want to double check it is actually that. And someone said, like, do we use it kind of like a control? And I guess there is an element of control there as well, to check that the thing we've sequenced does vaguely match the stuff that we already know about as well.
Yeah, amazing. Sorry, I probably jumped the gun then, but yeah, those answers are all correct. Yeah, we I guess in some ways it's probably more to do with what Deborah's answers was, but they're all correct in their own way. What they're saying, it's a control. It's there to help us align to the reference, because, and this is kind of what I was moving on to the next one in a very simplified approach. Essentially, what we're doing is, if I can try and show you, hopefully you can see me on the camera. So we have our reference genome here, and then the sequences. So it's going to do So you hear something called read depth, which is, you might hear, or it might say 50 times or 100 times. Basically, what that's saying is it will read it like this: bam, 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 or bam, 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 and it'll do that 100 times. And then what it does is, like the picture, that green picture, it says like there's a red line. It will kind of align the ones that it thinks there's a mutation. So we say we think there's a mutation here, and if they're all concordant, which is just a scientific term for they're the same, and they all kind of saying the same thing, that there should be a T here. Like the reference genome says there should be a T here, but all of the other ones are saying there's a C there. We know there's a mutation. Well, there's a base change there. Not mutation, but base change. So that's kind of how we're detecting variants and seeing how they are. And that's why we use alignment. So short reads is essentially taking those tiny pages, making sure that they're all kind of concordant with each other, to then see if there's a variant there. Hopefully that makes sense to everyone. I've tried to make it make sense. If it doesn't, I'm very sorry. And I will try and find a different way to explain it.
A nice analogy I heard for like the lining up things is like someone's taking a book and cut it all up and trying to put the pieces back together. And so that's sort of where the reference genome comes in, that you're not just doing it randomly. You've got like another copy of the book to line it up against. That really helped me sort of work out what all of that alignment was about.
Yeah, exactly. And it's just sort of, making sure we're at the right place. And exactly. I think that's a really good analogy for them. So this is kind of, again, this is a kind of a big step up from the thing before. This is kind of going in a little bit more into detail. It might be something more to do for like teachers or anything more kind of that level. But this is kind of what we call, this is the main important slide that if I was going to do a talk on library preparation, I would include. This is my gold standard slide, is what we're really adding. And really what we're adding is we're just adding these things called tags, and you probably would have heard that term in genetics. Tags is just really, you kind of want three things. So you want your primer. So a primer in genetics is just a more or less a start sequence. It's normally 6 to 8 bases, and it's kind of initiates the sequence, and that's kind of from the primer. And that happens in kind of DNA replication and things like that, where primer is there. And that's what these sequences, SP ones, those like orange and blue, like blue ones, those are the sequencing primers, so read one, read two. And they're the ones that kind of start off the process when we're kind of reading the DNA insert. And then we have these index ones and index twos. You don't really need them all the time. We only use them when we do COVID, because to save money, because scientists are cheap, we, instead of sequencing one sample at a time, which would be like £1,000 ago, I could just get everyone's and put them all into the same liquid. But so I know whose is whose, I need to make sure I have some kind of identifier that tells me whose is who. So these indexes are like little barcodes, where if I was to scan one, I know that this is person one. And if I scan another one, this is person two. Does that kind of make sense? So index 1 and 2 are like very unique. You know what? It doesn't really matter too much, but the idea is that these tags are there and they exist, and they're really just to help sequence kind of bind. So these last two, they're just there to bind onto the sequencing flow cell, which is the thing we put into the machine. It's just there to help bind, help us identify it, and help start the sequencing process. Those are really all you really need to know about library preparation, and just be aware of the term tags. It's something that they use all the time in industry. So this, again, I'll probably will skim over this really quickly and not go into much detail at all, because this is kind of where it gets in danger of kind of confusing people a lot, and this still takes me a while. It took me a while to get wrap my head around this as well, but really what you're doing is you get these like mini little wells, and you're essentially just each sample, so each different person's like DNA that we're sequencing, like kind of shotguns a hole, and they like, this is mine. And then they will, using something called bridge amplification, they will do these weird like bridge thing, like that. And they will just extend and they will amplify, and they will just do loads and loads and loads and loads of amplifications. And then that way, that's how we get those reads, those fragments. So it like basically copies itself 50 times, and that's how we can read it 50 times, because there's 50 of them that can read. That's how kind of it works for each kind of sample. And then once it's kind of amplified a bunch of times, it then can kind of come up as a straight line ready to be sequenced. So that's a very kind of overview of full cluster generation, but hopefully that kind of makes sense to you. And then this term, which I wanted to kind of introduce to you, maybe not understand it because it is still a difficult idea to understand it, but it's called sequencing by synthesis. And this idea that you want to, the reason we have two reads is because we can only read things in one direction. We can't read two lines at the same time. We've got to do one and then the other. So essentially what we're doing is we have our thing here. We want to just read the first one, and then it's going to do something called paired end turnaround, which is where it just flips, and then it just flips over, and then you read the other side. And that way you read both sides. But the one thing you've got to understand is it's not two different DNA. It's one DNA, but you're reading it from the start, and then you're reading it from the end back to the middle, start, middle. That makes sense? So that's kind of sequencing by synthesis in a broad term. So this is the NovaSeq. So this is my colleague. We're loading one of the machines. It's kind of what it looks like inside. It's pretty big. It's about half a ton, and it costs about £1 million. If you were to buy one now, I think Sanger gets them a little bit of a discount, but yeah, they're very expensive, but they're very accurate, and they get something like 99.99% of the bases correct. So it is very reliable, and the sequencing process is unique to the campus that we do. And Lumina use the same thing because we have quite a fleet of them here, but we do a lot of sequencing at Sanger, and that's kind of what we use it for. We have a lot of applications for sequencing. So one of the applications I use it for is for COVID. So what we look for is COVID gene. So what we're doing is we're sequencing all the genes, like the entire genome, which is, I think it's 30,000 bases or something like that. It's not too long. It's pretty short in that way. And we're just looking for different base changes, and we come back and tell the government. So we didn't find Omicron, but we found Omicron in the UK because there's something called the S gene, which is the spike protein, and we saw that there was like big mutations, like big ones, and there was like a micro deletion, so like a small deletion as well. So we could see that it was mutating, and that's how Omicron came about, because the spike protein had changed to a point where it was a different shape, and then it was attacking the immune system in a different way. And that's kind of what the application that we use it, that I use it in Sanger. But there's many different ones. I think there's lots of cancer genetics as well. We use short read sequencing and things like that. So lots of applications to it, but that's just one of them. So we're going to kind of move on to long reads, and something that probably it's the same kind of concept, but it's just longer, I guess. But a lot of people say, like, why bother with it? Short read seems good. You say about the high accuracy, you say that it's fairly cheap now, get the things done, things like that. But why bother? So one of the things that we do in Sanger is something called the Tree of Life project, which is where we're trying to sequence every organism in the whole of the UK. It's a very ambitious project, but long read really helps us sequence those genomes that we've never seen before. We don't have a reference genome. We don't have anything for that because we've never done it before. So there's nothing really to align it with. So these long reads mean that we can actually get the whole genome more or less in one read, or some cases we might have to do 2 or 3. And it also means that we can just do less reads to cover that gene. So it means that we can actually say with a higher degree of certainty that this 12,000 base long read that you said is pretty accurate. Now, when you're looking at small little things, you look at the small little things. You don't generally put it together. So because of that, you can detect like small mutations, but a lot of the time it's quite hard to visualize the structural and all abnormalities. So what I mean by that is like chromosomal abnormalities or things like that. So that's your translocations, or where like a bit of your chromosome moves to a different bit, things like that. So long read is really helpful with that, and whole genome sequencing in general is good for. You can only detect structural abnormalities with that. And one of the big things about long read is you can directly sequence RNA, which is a really weird thing, because in my job because we're sequencing COVID, one of the big steps we have to do is convert our RNA into DNA. Otherwise, our process won't work. So the fact that you can just do directly RNA is a big deal, and it's very kind of groundbreaking. So one of the ones we're going to kind of go through, and again, Oxygenable, I'm no expert, so I'm probably know as much as you guys after the end of this lecture, but I do know a little bit, so I'll try and explain as much as I can, but again, in a bit of a simplified format, just so you can understand. But it is a very interesting topic, so if you want to watch me, I'm sure Em will hand out some YouTube videos or things like that you can watch. So if you're interested, you can kind of do that and probably come back and correct me if I'm wrong.
I'm just going to pop a link in the chat box. Actually, we've got sort of like a little overview sheet that will go into a little bit more than you will just now, because I think Oxygenable is really, really cool.
Yeah, Oxygenable is awesome, and honestly, it's like one of the big ones. It's not quite as good as PacBio, which we'll go talk about later, but it's it's pretty big deal. Oxygenable has kind of come up and coming one, and it's kind of the one to look out for. So it's essentially, in a very simplified format, they have something called a nanopore protein, which in nanometers is 0.00001 meters. It's very small, and it basically creates a hole in the membrane where a molecule or DNA can come into contact with that nanopore, and it creates a ionic current because the nanopore protein is charged, and the DNA is also negatively charged. So when that comes into contact, you create an ionic current. And as a base comes through, it will have a different current. So, for example, this current might be an A, and that might be a G. And based on that current, you can essentially work out what base is what. And that's how Oxygenable works. And it's very awesome because you can, as you said, we can do directly RNA. We can do loads of stuff with it. It really is amazing. And the only thing that to kind of point out here is it's not as simple as saying like, oh, well, it knows what base it is because of the current. You can have like 6 bases at a time in the nanopore membrane making that current, and it will just keep flowing through. So if you have 6 bases, you have 4 different possibilities. It could be that's around 4,000 different states. It could be like the current. So a lot of it is kind of a bit of guessing as well, because not guessing, but like when you're kind of measuring that current, you can't always work out exactly what bases there are. So it's not, insanely accurate, but it is, it does give a very good answer. So nonetheless, nothing to take away from, but it is something to know as well. And again, when you do your reading, the nanopore proteins are kind of unique as well. So I know that they kind of, why it's called Oxygenable, they kind of like patented, they make different nanopore proteins that are differently shaped and stuff. So it's very cool. And it's something to definitely look out for. Now, not Oxygenable is, so I kind of highlighted the ones that I think that are bad in red and the ones that are good in green. So Oxygenable is awesome because it can do long reads, and when I mean long, it can do up to like 2 million bases, which is insane. Like that is really awesome, and it's also very portable. There's actually an Oxygenable you can get, which looks like this, and you can literally take it out on the field with you and sequence, a plant or something. Sequence something that's not, terribly large, and you can sequence it, and you can do all of the bioinformatic analysis or the variant reference alignment on your laptop on the field when you're in the field, when you're in the jungle or something and you're doing research work. That is insane. It's really kind of, you're not having to lug that 0.5 ton sequencer that we have in our labs. You can just carry it in your pocket, which is great. Now, the one thing to know with Oxygenable, which is kind of what I was saying before, how it's a little bit behind PacBio, is this idea that it's got a very high error rate. Not very high, it's 10 to 15%, but it can be biased as well, depending on what we're sequencing, if that makes sense. So they're not good error rates. So it means that the errors will kind of come in chunks. So sometimes it can be a little bit inaccurate. So a lot of people in industry kind of tend to use PacBio. So I know in Sanger, we are doing stuff with Oxygenable and waiting kind of their release. Oxygenable is releasing new kits and new different kind of methods and updating their things to kind of make it up to that level. But PacBio is kind of the standard in Sanger, at least. Maybe not everywhere, but PacBio is kind of what they use now. It's the same concepts, really. Let's not get too complicated about it. It's really just these tiny array wells, kind of like with Oxygenable, but they have DNA polymerase in it. And essentially it's just taking pictures of these fluorescently based pairs. So there might be some like dud, so like fake base pairs if you like, and it will take a picture and it will just be blank and it won't like have any color. But then when the DNA polymerase actually grabs a base, so like a G, it will release a certain color, and then based on that color we can work out what it is. Now, one of the reasons why PacBio is good is because it has these tags. The things I was talking about are loops, and it kind of makes this dumbbell shape. And this loop just means that you can actually sequence the DNA. So it's called a consensus region, but you can essentially read something, and then it can go around the loop, and then gets read again, and then goes around the loop and gets read again. And you really having this, it can be read many times, and that's how we get this accurate read. Because what you actually get is you get this. What I was saying before is you kind of have, someone's pointing my finger. You get a read where it's gone around so many times that actually you end up with a very, very accurate read. at the end of it. So you can actually be pretty certain it's almost as accurate as those Illumina ones we were talking about. That one of the bonuses that short reads have. So yeah, PacBio at the moment is kind of in the lead. Now PacBio does have a high error rate, but it's random. So because it's random, it's not biased. It means we can say with the results from PacBio have a higher degree of certainty, and it's partly because of that loop, because it can literally keep looping around, and because of that loop, it kind of has these reads that are all kind of saying the same thing. So then you can say with a higher degree of certainty that this is definitely the read. Now, PacBio doesn't do it as high. It's only 100,000 compared to Oxygenable, which is like 2 million, but it's still quite long. It's much longer than Illumina, which I think the highest you can go with Illumina is like 600 per fragment. So yeah, it's still much longer than Illumina, but not as high as Oxygenable. But kind of like a good summary here is try to kind of, take the different, like kind of the main aim of this was to see what's better, long read or short read. So even if you don't know anything, if you didn't really understand any of this stuff, which I'm sure, based on the answers and everything, I'm sure you all probably know more than me, but if you can only just understand or take away just this slide from the presentation, that means you've learned more than enough than I wanted, really. So long reads are good because you can do more than 10,000 base pairs long sequencing. You can sequence new and challenging genomes. As I said, you can see structural variants and things like that. Downside is it's a bit harder to prepare and it costs more money compared to short read. Short read is good for identifying those small deletions, so micro deletions and micro insertions. It's very accurate. And you can sequence up to 600 base pairs long. It's kind of the difference in lengths and why we call short and long. And just kind of the last one, this is how I like to think of it. Now, this isn't necessarily correct, even though it is. But this is the way I like to. Long read is, short read is taking these puzzle pieces, makes it a lot easier because we know what puzzle pieces we have now. But long read just gives us larger puzzle pieces to put. And obviously we can agree long read is easier to put together than short read. But one thing you have to remember as well, they both have their place, and not one of them won't replace the other. But they'll both need to be there. So questions. So thank you all for listening. Hopefully that wasn't too boring or too complicated at all. But yeah, any questions?
Awesome, thank you. We've had a couple already, but if anyone has any questions, pop them in either the chat box or the Q&A, and I'll pull some out. We had a really good one earlier, sort of around the structure of DNA that you were talking about. Mm-hmm. So someone asked if there are proteins. If DNA makes proteins, but then DNA is covered in proteins, where did those proteins come from? If they're there before the DNA, I thought it was just a really, really good question.
So this is the idea. Let me just see if I can go back to the slide.
So I think it was, your.
So is this? I'm pretty sure I know which slide you're about.
Slide 10.
That was a concept that I didn't explain as well. So is it this idea of how these are here? How can we express the gene if
the other way around? That if we need the DNA to make the protein, but the DNA is trapped inside protein, where did that protein come from? I presume it's to do with mitosis and that it came from the cell that made the sort of cell before, but I hadn't sort of thought about it like that.
Oh, okay, so sorry. Sorry, I can't see the question myself. So I'm sorry, I'm being stupid. So you're asking if
where did the histone? What makes the histones?
Come from? Okay, so unfortunately, I don't know that question, but that's something that I can definitely kind of find out and get back to. But it's kind of really
I presume
weird.
It's in mitosis, right? That as a cell divides and makes a new cell, it copies everything, and so.
So the histone proteins, I'm not really sure where they really come from, but if we talk about DNA replication, that kind of, it will double, and that's how new DNA gets made, is mitosis, where it will double in DNA and then decrease, and that's kind of how new cells and stuff are made. I'm assuming the histone proteins are definitely made from something. I'm assuming that will come from, it won't come from a gene, it will come from something else. So I'm assuming that's how it'll be made, but I thought the question was, how do we express our genes if they're already tightly wound together, which is another thing. But no, Sorry, that wasn't a very good answer.
No, it's great. It's I hadn't ever thought about it as a question. So well done whoever asked that, because you stumped the both of us. So we'll try and look into that. But I'd imagine it's in like the sort of, if you think about the cell before the cell, it will have made them. But it's a really good question, because you kind of get into a chicken and egg, that you need the DNA to make the protein, but the protein's around the DNA. So that was a really good question. We had a question linked to sort of things that cause mutation, and a more amusing question of, if we ate something that was mutated, can that mutation pass on to us? And they were thinking about the three-eyed fish in The Simpsons, which I also enjoyed those episodes.
Kind of, yeah, it could do. Probably not so, like, if you had the three-eyed fish thing, you wouldn't get three eyes. But I mean, like, you can have things like, if so like mercury poisoning, things like that. So you can kind of, if you eat things or ingest things that can damage the chemistry of our body, then yeah, you can get mutations based on that. So
and in terms of like actual food, that like eating another animal and whether that can cause mutations, on the whole, it's no. But if you look into like things like mad cow disease, there's a sort of specific type of mutation that causes a protein called a prion, and for reasons that we don't know, it can like transfer that change onto the people that eat it. So if you're based in the UK and you've heard of sort of foot and mouth disease or mad cow disease and sort of really big public health problems, that is as near to like eating mutated stuff and becoming mutated as that we get. And that's why there's always like massive safety precautions put in place when things like that break out. So prions are bizarre.
I think as well with the only thing I can think of right now that where you eat something and you get mutated is probably not mutated, but you can go crazy. So you can't really eat human brains or brains of similar kind of close descent. So if you were to eat a human brain, you'd eventually you'd go insane. And it's something to do with like the way what's in the brain when you eat it, it affects your chemistry and then makes your brain go crazy essentially. So that is the mutation in itself. And maybe not fully relevant to the question, but you can also get parasites where they'll live so there's one with mice, so they will live in a mouse and they want to be in a cat, but they don't really know how to get there. So what they do is they will make their way up to the brain, infect their brain, and then tell their brain that they are now sexually attracted to cat's urine. So then the mouse will then actively look for cat's urine, and then the cat will then obviously find the mouse, eat the mouse, and then the parasite will then make its way to the cat's brain, and that's where it will live out the rest of its days. And so there are things like that, but yeah, I don't know if that's entirely relevant to the question, but just something new, I guess. Sorry, was there another question? Oh, I can't hear. Sorry, Em, I can't hear you.
Sorry, I muted myself, which doesn't help, does it? We've got one sort of we'll take one last question before we wrap up around the COVID genome, because I know that's what you sort of work on at the moment. So someone's asked, when you sequenced the genome of COVID-19 and you did identify mutations such as Omicron, did you try and repair it using things like CRISPR or genome editing? I thought that's just a really interesting question. Oh, it looks like Sheree has frozen. How exciting. I'll pick up that question. So at the moment, in terms of specifically about COVID, we very much just sequence it and then, as Sheree was showing, work out if there's been changes and then basically pass that data on, and we don't have sort of involvement in that. CRISPR isn't really used for editing things like COVID. It's a really interesting piece of technology, and if anyone wants to know more about it, I'll stick a link in the chat box if my website wants to load. I'll stick a link in the chat box about CRISPR, but essentially CRISPR tends to be used more sort of in studying rather than in practice just at the moment. Sorry, we lost you for a second there.
Sorry, my computer just decided to die. So
I think we'll wrap up in a second anyway, but I was just saying we don't tend to use CRISPR to study, to like edit things like COVID. I guess we could use it to study COVID, but we're not trying to, I guess, repair COVID mutations. Awesome. I think we'll wrap up just there whilst everyone's Wi-Fi is still holding on. A massive thank you to you, Sheree, for joining us this evening. I do have a finishing slide that I will throw on the screen to round us out. Thank you for bearing with us for those tech issues there at the end. We will be hosting another one of these sessions next month. We'll be looking at parasitic worms. So if the sort of more gruesome facts that we were chatting through at the end appeals to you, then join us next month for that. The link is on the screen, and you'll get a follow-up email from the session about that as well. For anyone asking about the video recording, that will be available within the next couple of weeks on our website and on YouTube as well. So if you wanted to catch anything up afterwards, you can do so then. But thanks so much for joining us, and hopefully we'll see you again next month. And thanks, Rhea, for joining us this evening.
Everyone.